Great, thank you. I'm very delighted to be here today and be able to present in person. It seems like it was just yesterday, yet 100 years ago, where we were here in March of 2020 with the previous meeting. We've heard a lot about a vision for the exposome in a number of presentations during this meeting. And I'm an engineer in training, and much like a structural engineer would take a blueprint from an architect and make a building that doesn't fall down, I like to spend a lot of my time and research effort thinking about building a technological basis or foundation that will allow us to pursue the human exposome, or measuring the human exposome. So in my presentation, I'm going to cover three topics. I'm going to focus on defining measurement of the exposome from a practical perspective. We understand that the exposome is really considered the sum total of all of our exposures across our lifetime, but that's a very ambiguous and kind of almost impossible definition. So what are the ways that we can think of the exposome from a practical perspective and a way that we can actually apply measurements that allow us to characterize it? Next, I'll talk about how we're using high-resolution mass spectrometry to measure the human exposome. So I'll give you a little hint of what's to come. That's the technology that we're really basing our approach on. And finally, a brief discussion of how we can look towards the future and really start establishing the framework and the capabilities to measure the Human Exposome Project, or the Human Exposome Atlas, or MAP as Bob called it. So as we've heard from many presenters, prior to completion of the Human Genome Project, there was this anticipation that genetic testing would revolutionize our ability to predict disease risk. As we know, while there has been tremendous advances in genetics and our ability to use it within a medical setting, there's also a number of limitations, and we've really learned that genetics alone cannot be used to predict our risk for most common diseases. And a very good example of this was from a paper that was published from Steve Rappaport about 6 years now, where he looked at population attributable fraction for chronic diseases in monozygotic twin studies. And this population attributable fraction, which is essentially the proportion reduction in the prevalence of a disease, if you account for some risk factor, and in this case, since he was looking at monozygotic twin studies, he was trying to account for what the population attributable fraction is of these common diseases if gene or gene by environment risk factors are considered. And what he found was that for most of these, the proportion reduction in the disease, if you're considering genetics, is only about 20 to 30% at the most. So what this is telling us is there are a lot of other factors that contribute to our risk for disease, and many of these are arising from environment or lifestyle factors. So our current estimates suggest that really only about 5 to 15% of diseases are attributed to heritability alone. The other 85 to 95% are derived from a combination of our environment, lifestyle, and its interaction with the genome. So if we're only considering genetics when we're trying to evaluate risk factors for disease or predicting disease, we're really taking an unbalanced view of human health and disease. So to better account for this environmental and lifestyle components that can contribute to human health, we've really started embracing this exposome concept, as many of you have heard about over the many presentations we've had. And when we think about the exposome in its totality, really we can consider the exposome to include all non-genetic disease risk factors. These are things like ecosystem and psychosocial factors that contribute to our health, as well as physical chemical factors like temperature or air pollution or chemical exposure. So the question is, these are all very important environmental stressors and have the potential to contribute to our health and our risk for disease. But when we consider all of these in their totality, how can we ever really measure these to the level that we need to understand the human exposome. So in 2014, Dean Jones and Gary Miller, spent a lot of time thinking about this concept and the practicality of the exposome. And this led them to modify or essentially redefine the exposome in terms of cumulative measures of environmental influences and corresponding biological responses throughout the lifespan. And this is a key modification because it says that you do not necessarily need to consider every single exposure to measure the human exposome. Rather, we need to be considering the cumulative measure and the corresponding biological response to be able to characterize the exposome. And I'm borrowing this analogy from Gary, but he basically compared it to trying to locate mountains when you're on top of one mountain and looking over valleys and peaks where there's a lot of clouds hiding the base or the bottom of these mountains and the valleys. Do you necessarily need to know where every single mountain starts and ends to know where the peaks are? Or can you identify where the main elevations are by just looking over the clouds and identifying the peaks? And I think that's what we really need to think about and remember when we're trying to define exposome studies and exposome approaches to evaluating human health. We shouldn't necessarily bog ourselves down in the details of trying to measure everything we can. Rather, we should be trying to focus on what are the key measures of exposure and biological response that provide us with the most useful information. And so really, one thing that's critical in this definition is the recognition, and this was said very elegantly in the previous presentation, that in order for an exposure to have an effect, it first must be internalized within the body, because if there is no exposure occurring, and I'm mostly talking about physical chemical exposures at this point, but this can also be extended to psychosocial stressors and other forms of environmental stressors. But in order for an exposure to have an effect, there must be some interaction with the system itself, meaning that exposure must, if it's a physical chemical exposure, result in some toxicant or other exposure that's internalized within the body, and then that interacts with biological systems and causes biological effects within the systems itself. And thanks to the tremendous advances in omic technologies and other capabilities, we now have incredible capabilities to measure many of those biological effects and characterize the effects of those exposures. And so much of my work is really focused on measuring the chemical exposome. So I'm mostly going to be discussing that through the rest of the presentation. Again, the psychosocial and other social stressors that contribute to the exposome are incredibly important. They potentiate many of the chemical and physical chemical stressors that we experience. But a lot of our measurement techniques are focused on these chemical and lifestyle exposures from occupational and environmental sources. And that's really what I'll be talking about for the rest of the presentation and the technologies that we're developing to better measure these. And so to measure the exposome, one of the critical capabilities we need is a framework that allows us to perform omic scale biomonitoring of exposures and response relationships in human populations. We've seen tremendous advances in omic technologies that allow us to provide comprehensive characterization of biological systems. We have whole genome sequencing, genotyping, gene expression assays, methylation assays, proteomics. These are able to very comprehensively characterize the biological state of a system. However, our ability to characterize the environment is still largely relying on the technologies that we have been applying for the past 40 to 50 years. So the question is how do we bring measurement of the environment into the omic age and develop the analytical frameworks for universal screening of the human exposome? An example of why this is so important is some of our work that we're currently focusing on, which is using new analytical techniques to measure microplastics and plastic-related chemicals in biological tissues. And this is a figure from a recent paper from Helen Weisinger, where she attempted to account for every single chemical that is used in plastic and polymer manufacturing processes. And she was able to identify over 10,000 CAS numbers for unique chemicals that were associated with some stage of plastic manufacturing. These are only the chemicals that are actually used in the manufacturing process. They do not account for any transformation products that may be created or may occur during both the manufacturing process or during transport or uptake into biological systems, as well as, it's important to recognize that this is just for plastics. So if you can imagine extending these to all the different chemical industries and commercial industries, there's a tremendous challenge in being able to characterize exposures on the scale that they're occurring to us. And so the approach we've used is through development of methods originally created for characterizing the human metabolome as a unified approach for characterizing the exposome. So the human metabolome consists of all small molecular weight molecules present in a biological system. These include the endogenous metabolites that are critical for proper functioning of life and our biochemical processes, as well as chemicals that are arising from outside of your body. These include things like environmental pollutants, commercial products, and supplements and pharmaceuticals, and drugs. So within the metabolome, we have a single measure that allows us to both characterize internal exposures by searching for biomarkers of these exposures within the system, as well as a way of characterizing bioeffect. Because we can measure endogenous metabolites and the pathways, and in turn the pathways are originating from, we can actually characterize biological changes that occur in response to an exposure. So if you think back to that definition of the exposome originally proposed by Gary and Dean, where they said the exposome represents the cumulative environmental exposures and corresponding biological response, you can see that measures within the metabolome really represent a unique capability to measure the exposome. But we must remember that to measure the exposome, we need to do this on a scale that can allow us to systematically characterize the many exposures that we experience as humans. And so in order to truly embrace the complexity of the human exposome and develop the methods that will allow us to measure it, we cannot do this in a targeted manner where we are predicting a priori what we want to measure and what exposures we want to try and quantify. And so this is requiring us to adopt analytical strategies and study designs that allow us to incorporate untargeted or non-targeted measures of exposure. These methods allow us to measure many different compounds within a sample without necessarily knowing that they'll be there or trying to detect those beforehand. And then we can use data science and biostatistic methods to essentially tell us what the exposures or which biomarkers are important to our studies. So that's the goal. That's what we're hoping to accomplish with these technologies and platforms. But the question is, how do we actually apply this? How do we go from this hope of being able to characterize the exposome to actually measuring it in our human studies? And so in my research group at Mount Sinai, the high resolution exposomics group, our goal, or one of our goals, has been to really optimize high resolution mass spectrometry as a platform for population screening of the exposome, and also a cumulative resource that we can develop to study exposure-disease relationships. We've wanted to do this in a way that will provide an open and fair exposome analytical framework. We've heard a lot about fair data and its importance to advancing science and making data as available as possible so it can be reused and reanalyzed and taken advantage of. And that's something that we've built into the very analytical structure of what we're doing to generate our exposome profiles. So one of our main aims to accomplish this goal was to allow us to develop robust and large-scale profiling of the human exposome. I don't think it's going to be possible to continue using and applying methods that only allow us to measure 500 or 1,000 samples. We need to be realistically thinking about how we can develop analytical frameworks that will allow us to characterize tens of thousands of samples from diverse populations to measure the exposome. We also wanted to do this in a way that would be generating data that would improve accessibility for environmental health, biomedical, and clinical researchers new to untargeted exposomic research. There's a lot of interest in incorporating the exposome into many different types of studies, but there's often a knowledge gap and a steep learning curve that allows researchers to take full advantage of exposome data. So we wanted to make sure that we're developing a data framework or an analytical and data framework that makes the results as accessible as possible and provides opportunities for learning how to take full advantage of that exposome data. We've heard a lot of this in the earlier presentations, but our hope is also to do this in a way that will allow us to develop cumulative exposome disease atlases. And this is essentially a component of the human exposome maps or projects that have been discussed previously. These cumulative exposome disease atlases are really designed as a resource where we can aggregate multiple studies and multiple disease outcomes and essentially create a pan-disease exposome resource that can be used to evaluate similarities and differences in the exposome across different disease outcomes and populations. And finally, we're also trying to do this in a way that will allow us to integrate bioactivity and untargeted profiling to prioritize key exposures. We've heard a lot about that today with the zebrafish, which is a fantastic model, and we're also trying to think of other ways that we can do that with biological samples and more focused on in vitro assays. But I won't talk about that as much today. And so in order to accomplish this goal and our aims, there's a number of challenges that we've faced in developing comprehensive and robust exposome methods. Our first challenge is really chemical diversity and sensitivity. We know that our exposome comprises a tremendous range of different kinds of exposures and endogenous metabolites and drugs. And there's no way that we can use a single analytical approach to characterize all of them. At the same time, because we're performing exposomic experiments and not metabolomic experiments, we are also faced with challenges related to sensitivity. Many of the environmental chemicals that we hope to measure are present at orders of magnitudes lower than endogenous metabolites or drugs. So we need to make sure that the methods we've developed provide the sensitivity to actually detect many of the exposome biomarkers we're interested in. And so the way that we've approached this is to systematically optimize our methods and use multiple platforms to essentially develop omic scale biomonitoring approaches using multiple high-resolution mass spectrometry platforms. One of the great accomplishments, and this was in collaboration with the Hercules Exposome Research Center at Emory and Gary Miller at Columbia, was to use new high-resolution mass spectrometry technology that was developed by Thermo to allow us to expand our untargeted assays from more traditional approaches, which are based upon liquid chromatography and is more for non-polar or for polar molecules, to gas phase analysis using gas chromatography. And this allows us to characterize a much broader range of compounds with different physiochemical properties, particularly many that are related to the exposome. So through the development of these workflows that allow us to now perform untargeted assays using gas phase methods, we've been able to establish reproducible, systematic, and quantitative methods for exposome profiling of many of the environmental exposures we're interested in. We found that these methods are robust. They allow detection of both known and unknown environmental chemicals. And as far as GC goes, we've made the extraction methods relatively non-specific. They are still specific in the sense that they are geared towards volatile and semi-volatile compounds, but we've managed to design extraction and cleanup methods that are relatively universal to many of the compounds we're interested in measuring by GC. At the same time, we're incorporating improved and more comprehensive methods that are based upon liquid chromatography, high-resolution mass spectrometry methods. These have also been systematically optimized to allow us to detect many low-level exposures within our samples, as well as identify metabolites from most endogenous pathways. And an example of how these methods have allowed us to characterize that combination of both exposure and response is from a recent publication that was performed in collaboration with the National Cancer Institute, where we were trying to understand biological effects of occupational exposure to benzene. Within that study, we were able to use these high-resolution mass spectrometry approaches to identify both known and previously unknown metabolites of benzene within biological samples, and then link those to key alterations and pathways related to hematopoietic stem cells and their function. And so it really demonstrated the ability of us to use these LC-based methods to still characterize many of the exposure biomarkers we're interested in, as well as tie them to the key biological effects that have been linked to these exposures. But so it's really this combination and our ability to apply both untargeted GC and LC capabilities that allows us to provide systematic measures necessary for performing exposome-wide association studies. So many of our studies are now combining both LC and GC platforms, and we found that the combination of them gives us the sensitivity to measure many of these low-level environmental pollutants, which would be over here, but at the same time have the capabilities to screen and also detect many of the endogenous metabolites as well as drug and dietary metabolites. So this framework provides a basis that allows us to perform comprehensive chemical profiling to characterize the exposome. However, it doesn't necessarily give us the capability to take most advantage or full advantage of our data, as well as perform high throughput and large scale screenings. And that's what I'll talk about next. So the next challenge that we faced in development of these exposome level methods for biomonitoring is really identification of exposome features. I know many of you here have had the opportunity to work with or are familiar with untargeted data analysis. You know the frustration of designing a study, running the samples, analyzing your data, and being very excited about those results, and then coming to a situation where you have a number of very important features that are of interest to your study or outcome, but you have no idea what they are, and it's very challenging to characterize them. And one of the largest challenges we face with exposome studies is that oftentimes we need to rely on MSMS or dissociation studies to essentially give us a fingerprint of the features or molecules that we're measuring. This MSMS data, while not perfect, can allow us to make some predictions about the structure of the molecules that we're measuring. However, the challenge with MSMS is that we need relatively high abundant peaks to get good spectra that allow us to characterize them for the structure of those molecules. And as I discussed previously, it's very, very challenging or most of our exposome biomarkers are at very low concentrations, and that does not allow us to generate the high quality MSMS data that we need to determine the identification or predict the identification of many of our molecules. And so the way that we've approached this is through developing essentially a computational workflow that allows us to maximize traceability and usability of untargeted exposomic data for environmental health and biomedical research. We've essentially started creating layers of data that we can provide to collaborators that allows them to take as full advantage of the data as they're interested in or as their study really drives them to. These include providing multiple data tables from identified and high confidence confirmed metabolites and chemicals all the way through the fully untargeted data, which is basically all the information that we're capturing, whether they have identities or not. But the most critical thing, and I think one of the things that's going to be most important for advancing exposome research and making these cumulative resources is how do we get to a place where we have as many level 1 or identified compounds as possible. And so to support large-scale chemical identification, much of what we've focused on is establishing cumulative standard databases with automated confirmation and library building. And so essentially what we're trying to do is get our hands on as many analytical standards as possible, run them on our instruments, create workflows that allow us to pull the identification criteria from these standards and build databases that allow us to immediately pull these peaks out of our data as soon as they're run. To date, this has allowed us to confirm over 1,200 standards across our 4 LC modes, and additional libraries that are in the queue that we're planning to run include many other exposome and endogenous metabolites. So in total, this will give us the ability to have a database where we've confirmed over 7,300 unique chemicals representing exposures, drugs, commercial products, and endogenous metabolites for most metabolic pathways across GC and LC platforms. So this has been such a critical limitation in application of untargeted methods to exposome studies. We have all this data, but oftentimes we're limited to our least common denominators of what we can confidently identify. And so what we're hoping to do is build a resource, and this will be publicly available and open to any researcher who's interested in it, that has confirmation and identification information, including retention times, M disease, and MSMS when we can get it, for over 7,000 compounds. And this will allow us to support rapid identification of thousands of chemicals in exposome studies and greatly increase our ability to harmonize these and merge these across cumulative databases. But we also have to remember creating these identified databases and confirmed chemicals isn't necessarily, our end goal of what we're trying to do. We need to remember that oftentimes these unidentifiable features that we're detecting may be the most interesting or provide the most benefit to our studies. And I think it's really important to go back in history to really look at how one of the key exposures that many of us have studied were first discovered in biological samples and how they really first started becoming the key exposure that they're recognized today. And this kind of gives us a way to look towards the future because it helps us think of what we should be doing with our unidentifiable but relevant exposome features that we're measuring in our studies. And so I'd like to briefly just discuss the PCB story. And the PCB story is really about how a Swedish chemist named Søren Jensen in the 1960s first identified PCBs that were present in ecological samples. So Søren was an analytical or an environmental chemist who was working in Sweden, and he was using gas chromatography to try and measure DDE and DDT, two organochlorine pesticides present in eggs and birds and fish. And so he was running these samples, and he was getting very good peaks for DDT and DDE, which were two very widely used, or which was a widely used pesticide at the time. But he would notice as he was running his instrument that there would be these other peaks that would pop up later in his chromatogram. And he was using a technique called electron capture detection, which is specific to organohalogens, so chlorine and bromine. And so no other compounds should have really been showing up that did not contain either a bromine or chlorine compound. So he became very curious about what these were. He was noticing that he was repeatedly detecting them in different sample types. And as he moved up the food chain, the intensities of these peaks tended to get higher. So that was essentially telling him that they were bioaccumulating within the food chain. The other interesting finding was that when he looked at an herbivore, in this case, this was a sample collected from an elk, he could very clearly measure DDT and its metabolites, as well as dieldrin, which is another organochlorine pesticide that was widely used at the time. But you can see that in this chromatogram, where if you have a compound that's eluting at a certain time, it makes this peak here. You did not see any of those unknown but reproducibly measured peaks here. that were present in the other samples he was characterizing. Again, these are from a white-tailed eagle, pike. The white-tailed eagle eats the pike eats the young salmon. And so he continued investigating this and measuring it in multiple samples. And finally, he was able to get a sample from a white-tailed eagle that was very, very highly contaminated, and it had very high levels of this unknown peak here. And so he was then able to use what at the time was a new and still relatively crude technology called mass spectrometry, which you've heard a lot about in my talk already. And he was actually able to develop the mass spectrum for this PCB, and really, this brought recognition to the widespread recognition that these polychlorinated biphenyls were very potent and widespread environmental pollutants. But it's very important to recognize that this didn't happen overnight. He spent many years kind of studying these unknown peaks and characterizing them in different sample types before he finally had the opportunity to identify what they were. And we should remember that when we are designing our exposome studies and applying our exposome methods. Do we necessarily want to discard or think less of our features where we can't get MSMS for? Or do we want to start creating cumulative resources that allow us to note these features' existence and add them to databases and characterize what samples they're in? So while we might not know what it is immediately, we can start recognizing its importance across different diseases or sample types, and hopefully, once we recognize their importance, start thinking about ways that we can identify what these are. So I think this is something that we neglect a lot in our exposome studies. We tend to focus on what we can identify because it can be easier to describe what those are, but these unidentifiable features are just important and really should be a part of all the resources and data and results that we're generating. And the last challenge we face is this concern about low frequency and low effect exposures. And this is an example that was borrowed from genetics, where they talk about effect size and allele frequency. We know that many of the exposures don't occur uniformly across the population. Some are specific within certain populations, and others may have very strong effects and others very weak. And so we need to think about how we can account for these low frequency and low effect exposures, and also the importance of mixtures. And the way to do this is really through numbers at this point. We need to analyze large numbers of samples and really try to develop approaches that allow us to robustly measure the exposome in large populations. And so that's what a lot of my team has focused on over the past 2 years, is how can we develop fully automated open source methods that can be rapidly scaled across laboratories and provide robust approaches to the exposome. And so Brooklyn, Brismar, and Catherine, who are here in the audience, have spent a lot of time optimizing our automated sample prep methods that allow us to take one sample aliquot and prepare the plates and the necessary materials to run multiple assays on our samples for the exposome. They've been able to do this in a way that allows us to prepare approximately 84 samples in under 3.5 hours. Using one worker in the lab. So it's really allowed us to both scale up our capabilities, and the automated sample prep essentially makes us much more robust and independent of the operator. And we've also done this in a background that allows us to apply comprehensive and methods that really improve our ability to characterize chemical coverage. Now, every sample on the LC is being run using 4 different methods, and this allows us to detect many other exposome molecules as well as endogenous metabolites. And finally, we've created a data extraction framework that allows us to generate data tables from these large studies. Before, it's been challenging to reproducibly extract untargeted data from more than a few hundred studies. But now we're using cumulative and batch-wise alignment procedures that give us a reproducible analytical framework for generating internal exposome big data. And sorry, I'm running out of time, so I'm just going to rush through this real quick. And so finally, I just have 3 slides about looking to the future in the Human Exposome Project. So I think we are in a place where we can now start developing cumulative high-resolution mass spectrometry exposome databases that allow us to link exposure and disease in humans. This concept of the exposome map or exposome atlas should be our overarching goal and what we're aiming for. But I think in terms of walking before we run, we should start thinking of ways of how we can create disease exposome atlases. So rather than looking across populations and trying to get exposomes for everybody, can we start focusing on some of the higher prevalence or higher burden diseases and start accumulating exposome databases that allow us to characterize exposomes linked to, say, certain cancers or heart disease or diabetes. And these individual atlases, where we're looking at exposure similarities and differences across diseases and across populations with the same outcomes, will be components and pieces of building up that larger exposome map. And I think we're in a position where we can start this. There's been a number of advances and improvements that allow us to really start developing this framework for the Human Exposome Project. We have the technology, as you saw from the presentation. The scalable methods allow us to analyze thousands, if not tens of thousands of samples. We've made tremendous advances in compound identification and development of standard databases. We have the throughput and capacity. The study populations are there. We heard a lot about things like All of Us and ECHO and other cohorts, or very large-scale cohorts. The chemical coverage is there. The use of multiple platforms allows us to evaluate many different exposures and effects. And we have the data science for assembly. So these are the things that really allow us to move forward. However, there are still a number of limitations and obstacles we have to overcome. However, I think of these as cautions. These aren't stop signs or red lights preventing us from moving forward, but these are things that we should be considering with caution as we continue to develop this framework. And so we heard a lot about ontology. There are a number of limitations in ontology and linking information across different resources that we need for these cumulative databases in the Human Exposome Project. We don't have any good criteria for harmonization. So what do we consider a successful harmonization across data sets? We don't have clear criteria for standardization. How do we know if a method is performing appropriately to allow harmonization? Do we have the motivation? We've heard a lot about exposome projects and exposome maps, but we also heard from David Balshaw yesterday that most exposome studies aren't actually exposome studies. So do we have the motivation to undergo or undertake this challenge? We don't necessarily have clear metrics for success. Gary posted something on Twitter the other day talking about how they had finally completed the Human Genome Project, or were filling in many of the missing gaps. And we had talked about the Human Genome Project being finished in the early 2000s, and it was a tremendous success, but it was finished according to their metric of being finished. And that's what we need to decide for the exposome. What would we consider a successful Human Exposome Project? And finally, we don't have the resources, or we need to find the resources. What are the funding, and where will the support come for doing this? So in summary, improved measures of exposure and effect are needed for determining the role of environment in disease. The combination of multiple platforms, including GC and LC, provides a unified platform for exposome by metabolome-wide association studies of disease. And the new improvements in automations and methods really allow large-scale exposome epidemiology and biomonitoring. But this is another quote I'm going to borrow from Gary. Really, it's now possible to start developing the framework for large-scale exposome disease atlases and maps. And we must not let the perfect be the enemy of good. I think when we're planning these databases and atlases, we really can't let the details or the logistics or perceived limitations in current technology stop us from moving forward. I think the technology will meet the demand for what we're trying to do. And so we really are in a position where we can begin the Human Exposome Project. I'm sorry for running late. I apologize for that. I thought I had 40 minutes, but I must have misread the email. I just want to acknowledge my research group here at Mount Sinai, Catherine Dunn, Jingping Lin, Zoe, who's left for another position, Anna Roebuck, Brooklyn McNeil, and Brismar, who really are responsible for all of the work you're seeing on here. And thank you. And again,